Good afternoon. I hope you are still a little bit awake. We are going to talk about the chemical exposome with the eye of an epidemiologist from France. As my colleagues, we are going to start with a few basics. So brain aging is a lifelong process. In fact, the brain develops until up to 30 years old before having a progressive atrophy with aging, very progressive at the beginning, and cognitive performances also start to decline quite early in age. And some lesions may start to accumulate in the brain, vascular lesions, amyloid typical lesions, and other type of lesions. And this may lead some people to develop dementia in older ages. And of course, you all know that 2/3 of cases of dementia are attributable to Alzheimer's disease. And it is obviously a major public health issue. So prevention is really interesting because of these lifelong processes and also because treatment discovery has been a little bit complicated. Really, if we could find some preventive strategies to avoid these processes to accumulate, it would be really a good idea. So what epidemiological research has told us is that a brain which is better developed, more developed, with more numerous neurons, more connections, will have a higher resilience to cope with the lesions and potentially develop dementia later in age. So this is due to protective factors early in life. So that's an interesting aspect of research that is really ongoing, still ongoing. And if we could find also some preventive strategies that would avoid lesions to accumulate, that would also be very interesting. So really, to me, the key of aspects of dementia pathophysiology for prevention are the lesions, of course, but also resilience. And finally, a last very important aspect is time, because all these processes are evolving over the long term. And with epidemiology, we are well equipped to, with long-term cohorts to study this natural history of these processes during the life course. So the challenges for prevention are really to work on these time windows. I know my colleagues have introduced that already, but it's important to remind you that. And we have been studying a lot older ages because it is where dementia cases occur. But clearly, research has told us that risk factors assessed in midlife, earlier in life, are very important, even more important for brain health. And we also need to find the risk factors which are most important. And this is where the story is really complicated, because dementia and sporadic Alzheimer's disease is really due to a number of genetic risk factors and also a number of environmental risk factors. We are all citing the Lancet Commission, which lists 14 risk factors, which are robust enough based on existing epidemiological literature to be cited as the key preventive factors. But I personally believe there may be more, but it's a good starting point. In Bordeaux, because it's the slide where I really tell about our research we are doing here, we have been, I think, with the neuroepidemiology group in general, at the forefront of exposome research and dementia and Alzheimer's disease, thanks to our landmark cohorts. And this started in the 80s with the PACWID study, and then the 3-city study, a population-based cohort as PACWID, which was launched in early 2000. And then the Memento cohort, a clinical-based cohort later on, and even a new one, the B-Cube study, which was mentioned earlier. So this is really a nice set of cohorts dedicated to the study of brain aging and dementia and Alzheimer's disease that have been developed in Bordeaux. And we have collectively worked on many different aspects. So it's really a potpourri of studies we have been doing. We have been studying a lot nutrition, Mediterranean diet, cardiometabolic health, lifestyle in general, polyphenols, herpes viruses have been studied by some others. What else? Socioeconomic disparities are being studied also in the memento cohort, and more recently also some work on omic-based biomarkers and blood-based biomarkers in the memento also. So, of course, I cannot cite the work from everyone, but it's really a great group which has been working on that here. So I'm going to stop a little bit on this, one of these results that I like very much because it has been led by Jade Duffer, which is actually here in the audience, doing a PhD with me, and we worked on the first attempt to combine the lifestyle risk factors in a single risk score. That's one of these scores is called the Libra. It combines 12 modifiable risk factors in the form of the risk score. And we questioned whether the Libra would be associated with the risk of dementia and cognitive decline in the long term, and whether it would be moderated or not by genetic susceptibility, which was studied through the ApoE4 allele and through a genetic risk score. So we, in fact, it's difficult to see our slides here. We studied more than 5,000 older persons from the 3-city study here, and we calculated the Libra score at baseline in 1999, and the genetic risk score and the ApoE4 allele were here too. And we studied whether these scores were associated with cognitive trajectories in the following 17 years, and also the incidence of dementia, testing for the interaction. And in fact, the answer was we didn't find any interaction between the Libra score and the genetic risk, at least in these data. We found that having a lower Libra score decreased your risk of dementia, both in AE4 carriers and in non-carriers. And you can see here that the magnitude of this decrease is relatively similar, at least on a statistical point of view. And we found a similar size when studying cognitive trajectories. So we concluded that overall, thanks to lowering the Libra score based on this data, at least from a high Libra to a low Libra, we were able to prevent 12 cases of dementia for every 100 person followed for 10 years. And in fact, it's really a strong effect when you see the figures. And we concluded that this would suggest that acting on lifestyle and cardiovascular health could be efficient to prevent dementia in everyone, even in those with high genetic risk for Alzheimer's disease. So that's a pretty interesting public health message. So we could stop here and say, okay, let's work on the 14 risk factors and do preventive programs, and it's already ongoing and in the pipelines, of course. But I personally believe we should continue our research on etiology to improve the preventive strategies, because we still don't know exactly which factors are truly causal. We don't know which are the underlying mechanisms. Are they specific to neuropathological pathways, or more general and emphasizing, for instance, biological aging homeostasis pathways? We still don't know exactly which combination we should propose and is more efficient for prevention. The optimal time windows have been also cited. There might be some sensitive groups which may be more sensitive to preventive strategies based on genetic background or more general aspects, other aspects such as gut dysbiosis, immunosenescence, inflammaging, etc. And finally, I think we are all working here on what we call emerging factors, which are still emerging for the Lancet Commission, which are all these ubiquitous pollutants, for instance, to which we are exposed to, and for which we don't know yet a lot, or other factors such as viruses, and there are plenty others. So, we all agree, I hope, in this audience, that the exposome concept can really advance etiological knowledge and prevention of those pathologies. And this is what we have tried to explain in this paper that we published together with some of you. And the exposome concept, I'm not going to re-explain. I think it's okay at that time of the day. But here in this paper, we offered the exposome concept applied to neurodegenerative diseases, just to make it clear for the neuroscience community and neurology community. So in general, we can identify three main groups of exposome factors for brain aging. The first one includes psychosocial factors, such as educational level, social network, engagement in cognitively stimulating activities, the level of stress, etc. The other big group is lifestyle, nutrition, physical activity. We could put stress also here, sleep, medication use, etc. And the third big group includes all the chemical, physical, and biological risks, which is maybe less well characterized, and there are a lot of research programs on that currently. So what we are trying to do is to understand how these external exposures impact the biological fingerprints, and we try with biomarkers to differentiate true biomarkers of exposures from all the markers which are going to reflect the effect of these exposures on mechanisms and biology, leading in the end to the phenome, which is the manifestation of the disease, that we are going to study through clinical markers and also subclinical markers. And in our diseases, we are using neuroimaging and blood-based biomarkers, or fluid biomarkers in general, to better capture early brain mechanisms. So, in this paper, we made a focus on the chemical exposome, which is a part of the broad exposome, and we believe it's one of the important part of the exposome, which is maybe less characterized currently. And this includes, in fact, many different chemicals. I think one of you mentioned that this morning, that we are exposed to maybe more than 300,000 chemicals which are on the market, and really a very small fraction on that is well characterized. We don't know really what is the impact of these chemicals on the brain. And this includes things like metals, solvents, and pesticides, which have been actively used in the industry. All the chemicals which are emitted by combustion processes and air pollution. And finally, a big group of pollutants which are ubiquitous, synthetic chemicals which are added to consumer products, such as plasticizers, PFAS, etc. So this is a lot we are exposed to every day. And biomonitoring studies have showed us that we are all exposed to these chemicals. For instance, you have here data from the NIEHS in the US and the Esteban in France. And for all these families, you see that the frequency of detection or quantification of these molecules is very often 100%. It means that all this is circulating in our blood or urine, or is there, and we still don't know the impact of most of it on pathologies and on the brain in particular. So in this paper, we also made a literature review, scoping review, on epidemiological evidence on the chemical exposome and neurodegenerative diseases. We differentiated Parkinson's disease and motor impairment on the left, and studies on Alzheimer's disease or cognitive decline on the right. And we differentiated the literature on occupational exposures from the occupation, such as farmers for pesticides, and studies from the general population. And what we found is that clearly there is a strong literature on pesticides and Parkinson's disease for occupational exposures in farmers. I see Alexis Elbaz here is a specialist of that in the audience. And it's clearly considered as an occupational disease, Parkinson for farmers now. But in the general population, non-occupational exposures, the strongest evidence was for particulate matter from air pollution. And that's it. For all the rest, you see that there are small squares and light squares, means that there are very few studies and potentially inconsistent. So it means that all these ubiquitous pollutants we are all exposed to, we really need to develop more studies with epidemiological cohorts well characterized to understand more. And we also reviewed experimental work, and we found that indeed many different mechanisms were involved with exposure to these pollutants. You can see there are very general pathways such as oxidative stress or mitochondrial dysfunction, but also some specific pathways such as misfolded protein aggregation. And you see also that most of research is on persistent organic pollutants, metals, and solvents, but there is less around regarding more recent synthetic pollutants for which we have less data overall. So we clearly need to develop more research, and that's what we concluded. And I like this vision of the National Institute of Health and Environmental Science that I think that's David Balshaw that explained that to us a few days ago, and I like this approach, so I'm just resuming that here. The exposome is a concept, but how are we going to approach this concept with our studies? We are going to develop exposomics, which is in fact the application of the concept with our data. So it's about having the good tools for discovery, and we are going to leverage a lot on omics approaches, but not only. And it's exploratory by nature, at least in epidemiology. And finally, there is the concept of precision environmental health, which is about clinical implementation. So that's just to think and to have the concepts well defined here. So here in Bordeaux, we were at that point when the 3-city study was kind of old. I mean, all the participants were starting to die or being very old, and we wanted to implement a new cohort. And I wanted to create a cohort that would be able to operationalize exposomics and also detect early neuropathology. A cohort that would be able to have a good assessment of external exposures, but also molecular biomarkers to probe the internal exposome with correct biomarkers. I mean, we are all working on that, I know, but it's really a challenge when you want to implement that kind of study. And having deep molecular and imaging-based phenotyping to understand how the brain is aging at a stage when people are still not demented. I mean, it's before diagnosis. We are in the general population. We are also working on statistical methods to integrate all that. And do not forget socio-environmental disparities, which are modifying factors that can explain a lot of the differences we can find also across studies, across countries, etc. So this is where I'm introducing the B-Cube study, which in fact wants to make the link between the exposome, the genome also, because we are going to do that, link to brain vulnerabilities before diagnosis, and how this links to cognitive trajectories and could, in the end, improve evidence-based prevention policies if we understand all that. So it's not a very big cohort, only 2,000 participants, but we start at 55 years old to capture early stages of brain aging. And we have done more than 1,000 neuroimaging exams with interesting sequences, such as the one which assesses neuroinflammation, another one, the blood-brain barrier permeability. So we are really trying to phenotype well the brain of these people. They are living in Bordeaux, and we have done a very comprehensive biobank with blood, urine, stool, saliva, hair, nails, which I hope will allow us to really have a nice exposomics framework to make the link with all this wide phenotyping of the brain. So that's the strength of the cohort, and in fact it has allowed us to develop some transdisciplinary projects covering multiple exposome domains, in particular lifestyle and food, in which I've been involved in the past a lot, but also chemical exposome in general, biological factors, viruses, etc. And it's also used now here in Bordeaux as a kind of a reference model to try to implement also exposome research in clinical cohorts. And for instance, we have a large vascular brain health institute, which is dedicated to cerebral small vessel disease. And we are thinking about using the tools we are developing in B-Cube to do the same in the clinical cohorts with vascular disease, to have the same tools and different populations. I mean, I know it's a local initiative, but I think it's interesting to try to match population-based cohorts and tools with clinical-based studies. So as I was telling you, we are doing translational projects a lot currently. I'm going to give an example in a minute with an exposomics approach that really leverages this molecular epidemiology aspects, and I think it's really interesting. For instance, we want to question an exposome factor or a group of factors or the exposome at large, but maybe it's too much, and how it relates to cognitive trajectories and Alzheimer's disease. But we are going to work collectively, finding some signatures in the populations, of course, but validating mechanistically the findings with preclinical models. We can do also the opposite way. And that's what we are doing currently in the Exposignals project, which is a Horizon Europe project that will last 5 years. It has started 1 year ago. It is led by Véronique Perrier, who is here in the audience from Montpellier. And we have several objectives. Maybe I'm not going to list all these, but we start with screening in vitro some mixtures of pollutants that could really make a difference in terms of neurodegeneration-related pathways. Then we have an epidemiological objective, which I'm leading, that is going to make use of 4 different cohorts in Europe: the B-Cube study, the 3-city study, the Betula cohort, and the Constance cohort, which are complementary in terms of the age covered for brain health and also the period in which they were initiated. And it's important in the exposome field because we are not exposed according to the period to the same chemicals. So it's nice to be able to capture all that. And then we are going to, Véronique and their team are going to test some of the signatures and mixtures we can find in animal models and also in a model of neurodevelopment to address whether these mixtures would affect the brain very early in life. So I have 2 minutes left to explain you a few things about Exposignals. That's the overall epidemiology workflow we are using. That is going to adopt complementary approaches. The key thing is to use a multi-matrix workflow using blood, urine, and hair to capture different pollutant markers. And we are going to use targeted and untargeted approaches because we cannot capture all the different pollutants with the same technology. That would be too easy. So ICP-MS will capture metals, GC-MS-MS POPs, etc. So we are also using a new method developed at IRSET in Rennes. Arthur will give a talk on that in 2 days. We are going to capture non-persistent pesticides in urines with that. And hair is going also to be used to do more than 160 markers, which is also very interesting because hair is more reflective of the moderate term exposure than blood or urine. So I'm running out of time. I'm not going to describe these results on POPs that were published already. It's the first attempt to get a signature of dementia and cognitive decline that was published by Sophie Lefebvre-Arbogast recently. Overall, the results were quite null. It was a small sample, only 500 participants. So it needs really more data, epidemiological data. We found evidence of effect modification by APOE4 allele with which some evidence of deleterious effects for long-chain PCBs were found in E4 carriers, but the opposite in non-carriers. So this really deserves more data. I'm going to end here because Arthur is going to explain in details what we are doing with untargeted metabolomics in this project. And I will end with showing you the posters that are related to the ExpoSignal project. Some of them were already done just before. But if you want to hear more, you have some also tomorrow. And there is a talk by Arthur David on Friday, which is going to be very interesting as a follow-up. So thank you very much. And I'm happy to take questions.
Hi, good afternoon. Eric Toomey, my name, from Swiss Tropical and Public Health Institute. Thank you very much for the great talk and for sharing your insights with all those great projects. I was wondering, coming back to the very first part of your presentation where you showed the potential of preventing cognitive decline when you tackle it early, showing those potentially 12 cases avoided across 10 years in 100 people. Did you or did colleagues investigate which of those modifiable risk factors would be the best one to tackle first for policymakers in the sense of cost-effective, low-hanging fruits?
Yes, you mean the work on the Libra?
Yes.
Okay, so the hypothesis was here to combine all the risk factors and work on the additivity hypothesis. So we really took the score as a whole and questioning whether each increasing factor in the good way or in the bad way would linearly decrease or increase the risk. So that's really the hypothesis. When we extract the factors and try to tease out which are the most important, it's a different modeling approach. You are going to put all the factors in the same model and adjust each factor for the other. So it's really not at all the same question you ask. And when doing that, we found here that cognitively stimulating activities was one of the tops. But when you try to use that approach in another cohort or another country, you will end up with a different answer. So my personal feeling is that it's interesting to frame a global risk score like that and work on a combination and not trying to prioritize because it's going to be really different according to the studies. But we can discuss on that. It's yeah.
Yeah. So Rick Wychek, NIH. Quick question about rapamycin, calorie restriction. I mean, there are two ways promote longevity. Is there any connection with mitigating potential, mitigating dementia onset?
Yeah, that's true that in animal models it's really promising. Intermittent fasting, for instance, is really promising. With human data, it's complicated, especially as we age, because we have issues with denutrition and undernutrition. So it's really difficult to tell people who age to fast. And so fasting studies are really less successful with humans because of that. That's my opinion, but again, it's something which is really debatable. So what is your opinion about that?
Actually, I'm not an expert on this. I just thought it seems like rapamycin, there seems to be certain groups that are absolutely promoting this as a longevity factor. And just curious to know people who are actually studying this, whether or not there's any data to suggest that they can mitigate some of the negative effects. I mean, some people are promoting that this is not just in animal studies, but also in humans.
Yeah. I know some people are promoting that, but there is a denutrition problem, so it's
Actually, I have a question. I've been trying to think about how to phrase this, but when we're talking about Alzheimer's disease and dementia, we're talking about cognitive trajectories that were set over decades. And I'm wondering if it's realistic to think that a 5-year grant could ever capture whether or not it's effective. And I think that's a potential problem because it's, I think, quite honestly, funders have to think about maybe a 5-year grant isn't the best way to study this because I'm not sure you will see anything. Plus, 5-year grant is really 3.5 years of data collection. It's not 5 years. And I just wonder, and I guess I'm more putting it out there than asking a question, unless you think it could be captured in a 5-year grant. But I'm skeptical that it's even realistic to think it could be. And what do you
Well, the cohorts are built to be studied more than 5 years, of course, and we are collecting the data and keeping the data really to be analyzed after, retrospectively. But I would love to get a 15 years grant to continue my cohort. And I know that NIA people are around.
Jay, if I could just make a comment here. It's, I was talking about the All of Us cohort previously. I think this is another reason why we want to embrace these longevity cohorts rather than starting our own for 5 years.
An intervention. All of us is not doing an intervention.
Well, yet. So.
Thank you. That was great.